I'm going to talk about how the bacterium Caulobacter crescentus adhere to surfaces and form biofilm. And the way it does that is thanks to nature's strongest glue. So let me first tell you what a, what a biofilm is. So what we call biofilm is a complex community of microorganisms growing on a surface. Bacteria can form biofilm, but it's also the case of fungi or archaea. We are going to focus on bacteria today. Oops. So biofilm can form virtually anywhere. On any man-made or natural surfaces, you will find biofilm. So biofilm can be bad. When we think of biofilm, we can often think of infection, disease, or they are known to clog pipes or induce corrosions. But biofilm can also be good. You know, for example, in wastewater treatment plants, we use biofilm to treat the water. Or they can be good to turn milk into cheese, for example, or for bioremediation purposes. So it's very important for us to understand how biofilm works so we can prevent their formation when there is a place we don't want a biofilm, like for example, on a pacemaker when you are putting it in somebody's body. And we can improve the biofilm formation in places where you do want biofilm to do things. So usually, the way biofilm work, it's all biofilms start with a few single cells that will find a surface, and under conditions that are favorable for adhesion, those bacteria. Oops, do you see my cursor? Okay, so bacteria will adhere to the surface, and then the adhesion will trigger some physiological change, and the bacteria will produce adhesins molecules that can be polysaccharide, proteins, DNA, lipids, and a combination of all those. And this will form a matrix that will put the cells stuck on the surface. The cells will divide, keep growing, the matrix will grow, and you've got like this big mesh of bacteria and extracellular polysaccharides and other being formed. At some point, when the biofilm is too big, or if there is some condition that necessitates it, some bacteria can leave the biofilm, disperse, and they can find a new surface to colonize, and the cycle will start again. So the bacterium I'm working on, named Caulobacter, makes biofilm differently. So Caulobacter starts his life as a single swarmer cell. It's got it's motile, it's got flagellum, pili. And when he will hit a surface, this will trigger some physiological change. And the first contact with the surface will trigger the formation of the holdfast. The holdfast is the adhesins that will help Caulobacter to stick to the surface. Oops. So once the holdfast is formed, the bacterium will then differentiate into a stalk cells. It will grow like a stalk, and so it's actually pretty cute. And it will divide and to make a new swarmer cell. And the cell is being stuck to the surface only thanks to the holdfast. Then when you do that multiple times, you will start to have biofilm form with just single cells attached to the surface, just thanks to these adhesins. Here we don't have matrix, we don't have any complex cover on the biofilm. And before I continue, I want to stress out that the only way for Caulobacter to make biofilm, is thanks to the holdfast. Here we've got cells, wild-type cells that have been labeled in green, so we can see them. And we are looking at the surface from the top, and you can see that after a few hours, you've got a lot of cells attached to the surface, all these green dots. But however, if you make a holdfast mutant, you've got barely any cells, which means that if you don't have a holdfast, you can't stick permanently to the surface. This is very important because it will be for the rest of my talk. So now let's go back to our biofilm formation. We keep, we have those single cells keep dividing and and growing. So we've got a mature biofilm. And then at some point, new swarmer cells, they can disperse, find a new surface, like I was telling you before, and start a new cycle somewhere else. So the part we are going to focus on today is the biofilm dispersion. If we zoom on a biofilm, on a mature biofilm, so we can see that it's a complex population of cells of different ages and different cell shape, I will say. And we've got a dynamic population. We've got old cells that are attached, and we've got new cells being produced all the time. And those new cells, they've got basically two options. Or they can decide, or they can decide to join the biofilm and stick to the surface, or they can decide to swim away, disperse, and maybe find a new surface and colonize a new surface and make a new biofilm. So as we wanted to understand how we could control biofilm formation, we thought this was a good place to start. Can we find a signal that will, is there a signal that tell this cell, okay, join the biofilm or just disperse? Can we find a molecule to trigger dispersion? So this is what we first looked for. So for that, I knew that a mature biofilm, in a mature biofilm, we've got a high cell density. So if a molecule was to be excreted, it would happen at high cell density. So I just grew cells to a very saturated culture. Centrifuge, spin down, remove all the cells, filter sterilize my spent medium, hoping that if there was a molecule produced at high cell density that would trigger dispersal, I will find it. So I just took this spent medium, added to my biofilm, and see what happened. Of course, something happened. If not, I wouldn't tell you all that. And as you can see here, so this is the result of our standard biofilm assays. Here we've got a control. We didn't add anything. I set that up to 100%. And now you can see that if you add spent medium to the same biofilm, you've got only about 25% of biofilm made. So it means that in spent medium of a saturated culture, we can find a molecule that will trigger cells dispersal, that will tell cells in a biofilm, don't stay there, don't form a biofilm, move away. So very long story short, I managed to find the molecule that was in the supernatant that was responsible for biofilm dispersion, and it's actually DNA. And just for the side note, I'm not going to go into more detail, but it's specific for Caulobacter DNA. If you put any other DNA, it doesn't work. But so we know now that extracellular DNA excreted by Caulobacter is able to trigger cells dispersal, but we wanted to know how it worked. So for that, as I told you before, adhesion in Caulobacter is performed by holdfast. So what we did, we took some cells by microscopy. We can label holdfast using a fluorescent lectin that will specifically bind to some sugar present in the holdfast. So you can see holdfast here in green. We can label also extracellular DNA using another color, here red. And if we overlap, you can clearly see that both holdfast and eDNA overlap. So there is a good chance that eDNA will actually bind to holdfast. If you do the same thing in our holdfast minus mutant, of course you don't see holdfast, but more importantly, you don't see any localization of eDNA, meaning that eDNA is indeed binding to holdfast and not to anywhere else in the cells. So to sum up the beginning of this story, now it's pretty old, so I thought that it was not going to go into more detail about that. So we found in this first step that Caulobacter eDNA was able to inhibit Caulobacter biofilm formation, and that eDNA will bind and cover holdfast. We also found that eDNA binding will prevent the cells to adhere to the surface, both at the single cell level or later in the biofilm formation stage. And also, and eDNA also impair holdfast attachment to surfaces, even if you remove holdfast from the cells. And yeah, and as I told you, this mechanism is specific for holdfast eDNA. So then our next question was to understand where this DNA was coming from. So for that, we wanted to know if it was due to cell death or maybe like an active release mechanism like transformation in bacillus or something like that. This would have been cool, but unfortunately, this is not the case. So we look at, we monitored biofilm over time, and we can look at live cells and dead cells inside the biofilm just by using this specific stain. And here you will see live cells are green, dead cells are red. And we can look at the number of dead cells over time, quantify that. And we can also quantify the amount of extracellular DNA released in those biofilms at the same time. And you can clearly see that the amount of cell death in red and the amount of eDNA release are pretty well correlated, meaning that eDNA is just due to cell death and after cell lysis and release of the chromosal DNA from dead cells. It's not an active mechanism. So I was, yeah, pretty sad. I wish it was something cooler. It seems to, but maybe, you know, this is not just random cell death, but it will be something like a cell death mechanism, a programmed cell death mechanism. This could be cool too. So one of the systems that bacteria use as programmed cell death is toxin antitoxin. There is plenty of other types of programmed cell death mechanism in bacteria, but today we are focusing on this one. So basically, toxin antitoxin systems are very widespread in microorganisms. You can find them in almost every bacteria and archaea around the world. And they're always made, it's a very basic system. It's composed of two proteins. So you've got a very unstable antitoxin protein being synthesized at the same time as a very stable protein. So you can imagine that when the, and they are in an operon, sorry. When the operon is expressed to a normal level, I will say, you've got as much toxin and as much antitoxin being produced. The toxin will bind to the antitoxin, and the bound complex will, in this bound complex, the toxin will be inhibited and the cell will be happy and live. However, if for some reason the expression of this operon is changed and the equilibrium between the amount of toxin and antitoxin is broken, as the antitoxin is very unstable, they will be degraded. The toxin being very stable will stay in the cell. It will be free, and a free toxin is bad news for you. You will die. So in Caulobacter, there are now 14 toxin-antitoxin systems that have been identified. They haven't been characterized, but identified. And so far, we are just going to talk about 8 of them. So, what we did, we made some mutant for each single of those 8 toxin-antitoxin systems, and we look at we grow them in biofilm, and we look at the amount of cell death, the amount of eDNA concentration released in those biofilm, and the amount of biofilm formed. And you can clearly see, so here we've got wild type, like always. And you can see that only one mutant is different from wild type. This is the one we are going to focus on. And this is PAR-DE4. So in a PAR-DE4 mutant, you've got less cell death within a biofilm. You've got less eDNA release, which makes sense because we know that eDNA comes from cell death. And as a consequence, you've got more biofilm form because we know that eDNA will inhibit biofilm formation. So we decide to focus on this mutant, of course, and we wanted to know how this was regulated. So I'm not going to bother you with all the data. This is just the summary slides, like 3 years of my life here in one slide. But I could show that when we've got high oxygen, we've got a lot of expression of the PAR-DE4 operand. We've got a lot of antitoxin produced, a lot of toxin produced, but they will all bind together. So the toxin will be inactivated and there is no cell death. However, if you lower this level of oxygen, we've got less expression of this operand. So now we've got less of those proteins being made. The antitoxin being unstable will be degraded. You will have some free toxin lying around, and those free toxin will kill your cells. So now that we've got this mechanism in place, we decided to look what happened inside the biofilm if what we saw was true. So here what we are looking at, we grew biofilm on little plastic cover slips, and we are looking at 3 different locations in the biofilm. So we are looking at the top, at the interface between the air and the liquid, which is where there is the most oxygen available. So here it's like, here we've got the biofilm, and here is just the air above the medium. Then we are also looking at the middle of the biofilm, which we presume the oxygen level is in between. And we are looking at the very bottom of the biofilm where there is less oxygen available. And so we've got this, this way. So we fuse the promoter of our parent to Z, and we use a fluorescent X-gal analog that will tell us by microscopy the activity of the promoter under different conditions. And you can clearly see that here you've got more fluorescence, which means you've got more expression of PAR-DE4 than here. And the deeper you go in the biofilm, where there is less and less oxygen level, you've got less and less of PAR-DE4 expression. Now, if we look at cell death into those biofilm, so here it's the same, top, middle, bottom, a lot of oxygen, medium oxygen, low oxygen. You can see that wild type cells, we've got a lot of cells that are alive when there is a lot of air available. Green cells are alive, red cells are dead. And the deeper you go into the biofilm, where there is less oxygen, the more red cells you can see. So we've got more cell deaths. Here about, like at the bottom of my biofilm after 2 days, about half of my cells are dead. If you do the same experiment with a PAR-DE4 mutant, I think you can see the striking difference. It seems that they don't see that there is an oxygen limitation anymore. At the bottom of the biofilm, you've got as little cells that are dead compared to the top of the biofilm, where you've got a nice amount of oxygen. So those cells, they will die eventually, of course, but they are not dying because oxygen is limiting. They are going to die from something else. We don't know what, but I can see them dying after a while. So if we want to sum up all those results into a model, this is what I came up with. So here we've got a mature biofilm. For now, everything is fine. Cells are happy. So they produce the toxin PAR-E4 and the antitoxin PAR-DE4. Then the biofilm keep growing. So now we've got like a more populated biofilm. So the oxygen level will start to decrease. As the oxygen level decreases, we will have a repression of the PAR-DE4 operon, and we will have a degradation of the antitoxin, which means the toxin will be free. If we've got free toxin, we are going to have cell death. So some of the cells will disappear from our biofilm. And at that level, you know, oops, if I go back here, here we had plenty of bacteria. We didn't have a lot of oxygen available. But now here, by just killing a few cells, we come back to a more acceptable level of oxygen, I will say. But anyway, those cells are dead. Dead cells mean they will have eDNA release within the biofilm. The eDNA will stick to the old fast, which means the new cells, they can't stick to the old fast. They don't, to the surface, sorry. They don't have a choice, they have to disperse. And then when they disperse, they can divide, and the new cells can eventually find a new surface and grow a new biofilm somewhere else. So I think it's a very sophisticated mechanism, and this for 2 reasons. First, you know, like I think Caulobacter is smart. If I were him, you know, if I could sense death of my sibling, somehow that, you know, people around me will die for a reason, I don't want to stay there. I want to disperse, you know, I don't want to stick around. So, and by having these mechanisms that tell you like that, okay, you've got a molecule eDNA made upon cell death, and this tells you to disperse, I think it's a pretty neat mechanism. And also by just killing a few cells within your biofilm, first, okay, you've got the new populations that will go and find a better place, but you are also saving the rest of the colony, which, you know, if you keep growing and growing and growing, if you don't do anything, every cell that is attached will die. So you are at the same time spreading to a new place, to a better environment, and you are also preserving the colony. So I think this is a very, very nice mechanism. So of course, like the $1 million question is like, how this all work? And the answer is, I don't know. I'm working very hard on understanding like the entire mechanism and everything, but I don't know yet. But I am convinced that a way for me to find out is to understand more about the holdfast. And because if I tell you we need to understand more about the holdfast, is that we know very little about this molecule. Okay, holdfast had been discovered more than 50 years ago now. And everything we know about holdfast can be summed up on that slide. So you can see that we don't know a lot. So as I told you before, we know that holdfast is crucial for permanent adhesion. If we don't have holdfast, Caulobacter cells don't stick to a surface. Then, as it was on my title, and as Lorena said, holdfast is nature's strongest glue. We did some the lab, before I arrived, the lab did some biophysical measurement, and so far this is the strongest adhesion ever recorded for any type of adhesins, like man-made of bioadhesive. There is nothing more sticky than holdfast. Unfortunately, we don't know holdfast composition. We know that it's made with a polysaccharide, a series of polysaccharide, but we know it must be more than polysaccharide because polysaccharides are not that sticky. So, but we don't know anything else. Finally, we know that holdfast is very small. It's about 20 nanometer height. And here you can see like an SEM electron microscopy picture of Caulobacter cells with the stalk. And here you can see the little holdfast with a zoom. You can see that it's pretty small. And the fact that it's so small, we call it like a nanoscale bioadhesive. So when I talk about bioadhesive, what do I really mean? So for me, a bioadhesive is a bioadhesive. It's an adhesive that is found in nature, an adhesive that is made by a living thing to attach to stuff. There is a huge diversity of bioadhesive. You can find them, like in more complex organisms, like a snail or a starfish or spider, of course, like biofilm, bacterial biofilm. So there is more and more interest for bioadhesive, and there is a few reasons for it. First, because it can be a green alternative to synthetic adhesives, because synthetic adhesive, you need to be using harsh chemistry and like petroleum products and things like that, toxic things to synthesize synthetic adhesive. But for this type of bioadhesive, normally they are made by the organism is making them for you. So and it's less toxic. And as a consequence, so you've got a product that is less toxic and it's usually biocompatible, which means we could use it better for, like, for example, to stitch when you've got a wound. Instead of stitching, you could glue it or something like that with biocompatible adhesive. So there is a lot, like within the last 10 years, there is a lot of research done in bioadhesive. And we've got basically two classes of bioadhesive. We've got the adhesive composed of adhesins molecule, and the best example is a mussel bites. When mussels stick to rocks, actually, they are using this complex protein, protein adhesins that it's made of at least 6 adhesin protein. And the system is so well characterized that nowadays, researchers in the lab are able to express and purify those proteins and recreate the mussel glue. It's named CellTac, and you can buy it online. So that's good. Then another type of adhesin are the adhesive structures. The example is a gecko satay. So in fact, geckos, when they climb to surfaces, they will do that they will stick to surfaces thanks to little nanostructure on the tip of their feet. And you can see a picture here of a gecko foot. And in the lab, we are able to recreate these structures, like this complex architecture, and we can recreate it so well that we can actually make some gecko-inspired adhesives. based on those nanostructure. So you are going to tell me, okay, like we've got those bioadhesive, they're already characterized and everything. So why do we care about finding new ones if we already have what we need? Well, actually, there are problems with those adhesives, is that they are not really sticky. If you look at the adhesion strength of both the mussel and the gecko, you can see it's around 10, 15. And if we compare that to crazy glue, I don't know if in Uruguay you call it crazy glue or super glue, because in the US, super glue, yes, in France too. Okay, so let's go, sorry, it's crazy glue, but let's go with super glue. So you can see that if you compare it to super glue, the adhesion is pretty weak. But now, if you compare it to holdfast, I told you, holdfast is the stickiest compound on Earth. And holdfast is about 3 times more sticky than super glue. So I hope this is a good reason that, yeah, you will believe me when I tell you we need to understand why holdfast sticks so well because of that. And if you want to put things in perspective, if you take 1 square centimeter of gecko glue, you can stick this plastic figurine, which is pretty cool. But you know, now if you take crazy glue, super glue, I don't know if you remember the commercial from when we were kids. Most of you were probably not born, but when I was a kid, we had this commercial where they were just putting a drop of super glue on the helmet of this worker and they could stick it to the floor. So yeah, you can lift a man. But now, if we could cover 1 square centimeter surface with holdfast, we could lift a cow. In the lab, we actually don't lift cow. My dad thinks this is what I was doing for the longest time, but no, this is just based on calculation. But anyway, one day we will be able to lift a cow. So yeah, I hope I convince you that, yeah, it would be nice if we could characterize holdfast better to maybe use it as a novel bioadhesive. So, but of course, if it's not done, it's because it's not easy. And we've got a lot of challenges to solve. So first, as I told you before, the composition of holdfast is unknown. As we don't know what it's made of, we don't know how it's made. We don't know the synthesis pathway inside the bacterium. We don't know how Caulobacter make holdfast. Also, it's extremely hard to purify because it's so sticky that you will stick to everything. You can make it, you will stick to your tube. You can take a razor blade to scrape your tube, but then you will be stuck to your razor blade and it's handless. Because of its stickiness, we've got a very hard time to purify it. And also, it's made in such a small amount by the bacterium. Remember, it's 20 nanometer high. So that to have a decent amount to be able to work with, we will need like, I don't know, gallons and gallons and gallons of bacteria, and this is not feasible for now. So to try to characterize it, we needed a single molecule technique because, yeah, as I just said, no way we could have like milligrams of protein to use with conventional techniques. So as a single molecule technique, we decided to choose atomic force microscopy. So basically, atomic force microscopy is just a fancy scanner. So you've got, so it's this machine. And so you've got an AFM tip that will scan your sample. And you will know the position on your AFM tip in real time because there is a laser that will shine on the cantilever and reflect on a photodetector. So it means that the position of your cantilever will be recorded at any time. So usually AFM, so yeah, atomic force microscopy, I will say AFM for now. AFM is usually used for high resolution imaging. If you go side to side, if you scan side to side, so here we don't have, we are on the surface, so position of the laser on the photodetector is 0. And then, you know, when we will start to see some features and the tip will climb on those features, we will have like difference of deflection in the photodetector. And based on that, we can recreate a topography image, a 3D image of your sample. And this is what I've been showing here. Here we've got a Caulobacter cells, and here we've got a bunch of purified holdfast attached to a surface. So what's great with AFM is that there is no staining on or sample prep like that are sometimes very complex and can make some artifact. the only thing that you have to do is to have cells attached to the surface, and that's it. After you can scan them, no problem. And thanks to that, we can even image live events, which is interesting. But more interesting for this talk with IFM is not just a fancy microscope that can do nice picture. We can also actually understand some biophysical property of a material thanks to IFM. So first of all, and in the case of Allfast, this is what we are the most interested in, is that with IFM, you can measure adhesion. So this time I was telling you that to image something, we were going side by side and scanning our sample to have like the topography of the image. Now we can also go up and down. So let's see what happens when we go up and down. So right now we are like, our tip is going down on the surface. The tip being free, we like, and what we are looking here, like the green line, it's the deflection of the laser on the photo detector, which means the position of the tip. So yeah, the tip is free, so the deflection is constant until we reach the point of contact. So now, point of contact, if we keep pressing, we will bend the cantilever, so we will have a change in the deflection, and the change will be illustrated like this. And we keep pressing until a certain force. And now we decide to retract the tip. So what happened? We retract the tip. The cantilever will unbend slowly and both curves will overlap if everything goes according to the plan. And now if we've got a sticky sample, it means the tip will stay stuck inside the sample. And if you keep pulling, the cantilever will actually bend the other way. So you will have a negative deflection. You keep pulling, pulling, pulling until you reach the rupture point. Now the tip is free again. We go back to equilibrium. So it's a bit like, you know, when you step on a gum and and you try to remove your shoes, you will need some force to break the contact between your shoe and the gum. This is exactly the same. So, and based on those retraction curve, we can learn two things. We can learn the maximal force we need to retract the tip from the sticky sample. And also this area is what we call the work of adhesion. And I'm going to use those numbers more anyway. So now let's do that on Allfast and see what's happening. So in that setup, I made things a bit different just to confuse you a bit. We've got Allfast attached to the tip and not to the surface, but basically it's the same principle. It's just that for our experiment, it is way easier to do it that way. So at least we can test different surfaces, different conditions. This is way better for us to have a control environment, but same principle. So what we did here, we press on the surface, we just wait for some time, and the time we wait while we press in contact with the surface is what we call the dwell time. And then we retract and we measure the force we need to retract those samples, and this will translate to the adhesion force. And what you can clearly see here is that the longer we press here at that step on the surface, the more force we need to be able to retract the tip. So this means that the adhesion of Allfast is time dependent, and it's an exponential. It doesn't really matter on our case, but yeah. And this is true. We tested a bunch of surfaces, and this is true. This exponential increase over time, it's true on every surfaces. And just as a side note, we didn't find a surface where Allfast doesn't stick so far. You know, like, yeah. So to tell you that it really sticks to everything. And oops. So if you remember the little cartoon I showed you before, where the retraction curve were looking like that, here we've got our work of adhesion, our maximal force. And actually in our cases, only 40% of the curves look like that. And when it looks like that, it means actually that we just have one rupture event. When you pull your tip, Allfast is detached from the surface, or your tip is detached from the surface all at once. But the majority of our curves actually look more like this, where the rupture events happens in steps, which means that instead of having your sample being freed all at once, in fact, it goes more gradually, a bit like a zipper or something. And So what we did, we measured the maximal force for all those little peaks and we plot their distributions. And as you can see, we were extremely surprised to see that we had a distribution because frankly, at first I thought it will be completely random. And you can see that the distribution is actually multiple of 30. So it means that all fast is actually made of multiple adhesines that are arranged together, and each adhesin is 30 piconewton. And it's the sum of all the adhesin working together that will create all fast strong adhesion. Oops. Yeah. So of course we wanted to know what those adhesins were made of. So for that, we took our all fast, our tip with all fast immobilized on it. We just incubate it with different types of enzyme, and then we use our modified all fast tip and do the same kind of experiments. So here what we are looking at is basically the same force curve I showed you before, but now we are on a log scale. So you can see that on our controls all fast, no enzyme treatment here. You've got an exponential. You can't really see the exponential anymore, but it's the same curve. Yeah. And then what happened if we treat all fast with lysozyme? Lysozyme, we break down the polysaccharide. So here we can say that we've got a polysaccharide minus all fast. And you can see that adhesion is decreased about 60% when the polysaccharide are removed. So this was, you can say, well, 60%, this is kind of disappointed, but for me, this was a great result because it means that there is something else in Holdfast and that this is this something else that make Holdfast sticky. So I was extremely happy with this result. So if we treat Holdfast with a protease K in this case, where we will have peptide minus Holdfast. We didn't know if there were peptide in the Holdfast at this point, but to be honest, I tested all the enzyme we had on our freezer. So, and I just show you the ones that work. So when you treat Holdfast with proteinase, you can see that we've got a 95% decrease of adhesion. We are killing almost all the adhesion here. So it means two things. First, that there are peptides in the Holdfast. First time that we found something that is not a polysaccharide. And second, those polysaccharides are extremely important for Holdfast adhesion. So yeah, it doesn't look like much, but I was really excited. And then if we treat Holdfast with DNase, we can see that DNase also has an impact. This impact is pretty mild, okay, but it's happening only during the initial adhesion of Holdfast on the surface. But you know, it means that DNA molecules are also present in the Holdfast. So thanks to this first IFM study, we could show first that Holdfast adhesion increase over time. And it's due to the combined action of single adhesins molecule present in the Holdfast. Before we thought that Holdfast was a big homogeneous blob of something, but no, actually there is some organization inside the Holdfast based on those adhesins. And we also know now that there are peptides and DNA molecule present in the Holdfast. So I told you we were just looking at our retraction curves, but in fact we can learn more things from these types of data, and we can look at the approach curve. What happened when it's like now we are not trying to pull our sample away from the surface. We are just pushing on our sample that is stuck to the surface and see what happened. So if you do that on a clean surface, same as before, the tip is not in contact with the surface, so nothing happened until we reach the contact point. Now we've got a clean surface. If you keep pressing, the clean surface the clean surface being very hard and undeformable, you will have a maximal deflection and it will translate like that. If you do the same on a hard sample, so nothing happened until the contact point, and now your sample is hard but not as hard as the surface, so you will have the tip will bend but not as much on the surface, so you will have this type of curves. If you do the same on a soft sample, you will have even less deflection. It's a bit like when you take butter and you try to cut it with your knife. If you take butter out of the fridge, you will need more force than if you take butter that it's at room temp, the same. The tip will penetrate easier if the sample is soft. So these are our 3 examples, and thanks to the slope, we can learn about the stiffness and the elasticity of our material. The less stiff and elastic and more elastic the sample is, the lower the slope will be. But so now let's see, if you do those type of experiments, let's see what happened on Holdfast. This happened. And as you can see, even if you are not a biophysicist playing with AFM all day, you can see that it doesn't look anything like this. So first you have to be sure that you did all your control right. Then you have to convince your boss that this is true. This is the hardest part of the experiment. And then you have to understand and have these things make sense, because you know it's real, but you don't understand why. So if we take those curves and we think about it for a minute, here we know that we reach the surface. We've got the maximal deflection. It means we poke through the entire Holdfast and we are on the surface. Then we know that our Holdfast is about 20 nanometers. And so we know that the first contact point, we just go and before we measure that on each Holdfast, we measure the height of the Holdfast, so we know exactly where the contact point is once we know where the surface is. So on that case, this is here, and we set it to 0. But so we can orient ourselves like that. But it seems that actually the tip can sense something about 100 nanometers before actually hitting the Holdfast. So this was extremely puzzling for me. So I dug into a lot of literature and I found my answer into some old polymer science papers, which I'm not a physicist. It was extremely hard to read and everything, but they gave me the answer, so I'm thankful. So in those old papers, what they did, they took some polystyrene beads and grafted different sizes of polymers of different flexibilities and whatever, different polymers to make little beads with hair like that. And they look at how they will interact with the surface. They did that with a bunch of them, and they came up with this equation. So I'm not going to enter into any detail about those equations, because first, I don't have time, and second, I don't understand this equation. But if you fit this equation to my data, it fits, which means that actually I've got a model to explain my data, and which means that my Holdfast got a polymer brush. So I was always thinking of my Holdfast being like this little cloud of 20, 40 nanometers, but it's actually cloud with polymers. And those polymers are what the tip sense between the real point of contact. So I was extremely excited. So then, thanks to this equation again, we can extract 2 things that for a biologist are relevant. We can extract the length of the polymer brush and the density. So I took about 300 Holdfast, measured them all, and came up with the length of the polymer brush of 90 nanometers about, and the density being 10 quadrillion per square meter. I never used this word quadrillion before, so I'm excited. Anyway, sorry. Which means that 10 quadrillion per square meter doesn't mean anything, but it means that there is about 1,000 polymer per Holdfast. And I guess that's the reason why nobody saw them before, because they are kind of sparse, they are very flexible, so they were impossible to image. So yeah, and by having this polymer brush about 90 nanometer, when I told you that the Holdfast core height is it's about 40 nanometer, we triple the size of the Holdfast right now. So that was exciting. Of course, we wanted to know what this polymer brush was made of. So for that, I used the same enzyme I used in the previous experiments, treat my Holdfast with that, and redid indentation curves on those treated Holdfast. And you can see first that when you treat with lysozyme, you don't change the length of the polymer brush, or a bit, but it's not significant, oops, I lost my cursor, sorry. Which means that the polysaccharides are not a big part of the polymer brush. And I've got other experiments that I don't have time to show you that shows that, yes, the polysaccharides are actually part of the core of the holdfast and not present in the brush layer. However, when you treat your holdfast with proteinase or with DNase, you can see that you've got a reduction of the length of the brush layer. You also have a reduction in their density. I didn't have time to show you that. And so this makes us think that the brush layer is composed mainly of peptide and DNA molecule, and maybe of something else that we didn't find yet, but this is it. So on that, I would like to stop there. And I hope I convince you that holdfast, at first we thought that, yeah, holdfast was just one entity, but in fact, it's a complex multi-component layered structure. It's composed of a stiff core. I didn't have time to show you, but yeah, if you look at the indentation curve and you look at the stiffness and elasticity data of this core, it's very stiff. And so we've got this stiff core covered with a very flexible brush layer. So what we think is happening is that here you've got your cells with your holdfast. Now I had to modify my figure because holdfast got these polymers lying around. And the first contact between the holdfast and the surface is actually made by the brush layer. And if we zoom in, we think that this polymer brush will actually interact with the little holes and gap inside the surface. We know that because we've got better adhesion on rough surface compared to very smooth and atomic flake surfaces. So, and once the brush layer makes the first contact, we know, you don't have time to show you that again, that the brush layer will compact over time. And as it compacts, it will bring the holdfast core closer to the surface. And this is what will make the adhesion very strong. And now you understand why the adhesion is time dependent. So of course, we've got still plenty of things to learn. We need to understand how this whole mechanism works at the biophysical point of view. We also need to know which components are important in which step and how do they contribute to each step. We need to know if there are more components than DNA, peptide, and polysaccharide. And also, I told you there are peptide and DNA, but you know, this is pretty vague. I still don't know what is actually those peptides and DNA because, yeah, they must be specific ones. I don't know which ones they are. And once I will be able to identify them, I need to understand how they are synthesized, because if I want to be able to recreate holdfast in a tube, I need to know all that. But, and to finish, if we want to go back to our bioadhesive poster child, I will say, the mussel and the gecko. So the mussel is using adhesin adhesin's molecule. The gecko is using structure, but if in fact it seems like holdfast is using the best of both worlds, is using adhesin's molecule at the same time as it's using structure. So maybe in fact it's the reason why holdfast adhesion is so strong. It's because it managed somehow to evolve to make, like, to use both types of adhesins. And on that, I would like to thank my lab. So first my boss, Yves Brun, that allow me to do whatever I want in the lab and any kind of crazy experience I want to do. And so we were in Indiana, as Lorena said, and we just moved to Montreal last year. And in fact, the entire lab moved from Indiana to Montreal. So this is the lab. And also, all those experiments were done in Bogdan Stragnia lab at Indiana University because I was using his IFM. Now I've got my own, but it's not set up yet. And on that, if you've got questions, I'm happy to answer.